All right, it's my pleasure today to introduce Ms. HollyLane. Holly has been with me for a master's student, my third, for a little over a year and a half. She set an ambitious goal to complete her master's degree in a year and a half, and she's on target to do that. She submitted her first paper at Phenome Journal a little while ago, and has been doing some challenging reviews. What she's going to present today primarily is in her second topic. Also mentioned that she is a very proud Washington State alumni, and supports the Cougars anytime she can. And I think she really misses the temperature of Washington State. So I think when she finishes, she's planning on going back to Washington State to embark on a new career there. So Holly, please.
Thank you very much. I will say Pullman got some snow this morning, so I'm not missing it so much today. But the heat's been a little bit of an adjustment for me. So thank you all for being here. Like Seth said, I'm going to be talking to you about sort of my second project, and I'm still generating data for this. Still generating data for this as we speak on the computer. And so it's about looking at sort of the trade-offs between accuracy and throughput, which to our knowledge, really a simulation hasn't been used to do this directly up till this point. And so just to kind of give you a very general overview of the trends in plant breeding over time, plant breeding really started over 10,000 years ago when we first started making improvements to plants based on what we could visualize. So early phenotypic selection, so that was moving the ancestor to Ascente, to maize, and improving crops for our use. And then over time, with the movement of the scientific era, the rediscovery of Mendel's laws, when we really started to think about what was going on underneath what we could visualize. And so for corn, this is when we'd had the time like the hybrid era and things like that. And then it wasn't really until the genomics era where we've been able to see those things that before we could just sort of think about. So heritability. And now I'm sort of arguing that we're moving into a new era of genomics. So along with this genomics era, we've been discovering that we have a lot of phenotyping bottlenecks. And one thing I want to point out is I don't think phenomics technologies are going to replace genomics technologies, just as we have never really given up our visualizable phenotyping of plants. Each era has sort of built on the next, and so I think a lot of these phenomics technologies will build on genomics and complement them. And so what is high-throughput phenotyping? So I like to think of it as either increasing the number of observations that you're doing, the dimension of observations, so either spatially or maybe even adding a temporal aspect to your measurement where it was formerly a single point trait, and then sort of removing some of this human bias in these measurements as well. And so the question we really are asking in this project is quantity or quality more important in a breeding program? And what thresholds are making these differences? And so to get a little bit into the kinds of types of high throughput phenotyping we're talking about, so we have unoccupied aerial systems, things like drones, fixed wings, and these are really, really fast for data collection. So a Rice study estimated that while they could manually measure plant height for 45 plots in an hour, the drone was collecting data for 3,000 plots in an hour. So that's just the kind of throughput we're really talking about. However, I will say the caveat there is once you fly over the field, you then need a data extraction pipeline, which is going to require less human labor, but the human labor is going to need to be more highly skilled. Another thing is you're sort of increasing the spatial dimension. So now you're phenotyping the entire plot as opposed to just a couple of what you're deeming to be representative plants in the plot. Like I said before, this is minimizing human labor, though the labor is going to need to be more skilled, at least at this point in time with where the technologies are at. So also there's near-infrared reflectance spectroscopy, and this is a very highly precise measurement, so it's actually considered to be in some ways more precise than doing wet chemistry, though it does need to be calibrated with wet chemistry, but it's also very, very inexpensive. So we estimate that while it takes about 40 cents of labor per scan in the NIR, you're looking at anywhere from $11 to $33 for wet chemistry based on how extensive you want that analysis to be. So again, just the kind of throughput we're really talking about that can be increased. And I also just wanted to point out that the usefulness of NIR is starting to be recognized as expanding beyond just the traditional compositional traits. So there's a recent paper looking at phenomic selection using NIR in place of something like molecular markers, and that's also going to increase the cost significantly as well. So why would we simulate data when Dr. Murray and I are clearly having so much fun in the field collecting real data? Well, the main thing comes down to resource conservation. So you're looking at taking a couple of hours to do a simulation versus a couple of years, and that's going to involve less labor and, of course, then less money. There are also some advantages to doing a simulation, and a lot of people actually argue that doing an experiment sort of in silico is really a key step before you can move forward in knowing if your experiment or your method is going to work. And this really comes down to the fact that when you simulate a data set, you know what that truth is, and then it allows you to assess if your method is capturing that truth accurately or not. So our objective for this was to simulate data, both genotypic and phenotypic, and then to compare different scenarios where we measured, where we varied the error rate, so different measurement types, the number of environments we looked at, and then the sizes of the population. And then we wanted to assess how well we captured that truth that we simulated in the beginning. So just the workflow for the simulation, we started with a Hardy-Weinberg population, so you can think of that as an F2 with the allele frequency set right around 0.5. I used 100 SNPs, and we gave them a geometric distribution. So what this means is about 1/3 of the SNPs had no effect on the trait. The other 2/3 did have a varying amount of effect, with most of the effects being small. This was a 300 individual population. We then advanced it and selfed it once to create an F3, and then forced homozygosity like you would with a double haploid technology to create a recombinant Ig-Bren-Line population. Then we added some phenotypic effects, so an effect of each environment, each rep, G by E, and then of course error, which I'll touch on a little bit more in the next slide. And then I designed modeling loops where it was subsampling to vary population size, the number of reps, the number of environments, and then the varying error level of each measurement type. And then we loop this over and over and over, and this is what's still running. Each loop takes about 2 hours, and so if you want to do something like 50 iterations to feel like you're really capturing the mean, that's a significant amount of computing time. So the error rates were really dependent on what we were looking at for different measurement types. So the manual measurement, for the purposes of this project, we treated as the most accurate method. And so for this, I generated the error variance to be proportional to the genetic variance. And then each high throughput, or H2P, measurement we increased the error variance relative to the genetic variance. So 2x means that the error variance is twice the size of the genetic effect, and so on and so forth for 5 times and 10 times. And so the first method for assessing our sort of truth capture is we looked at best linear unbiased predictors, or BLUPs. And so we put this information into a model, these phenotypes that we've generated, and then we asked the model to give us sort of the best estimate for what it thinks the true genetic height of that line would be. And then we assessed the closeness of that prediction to what we simulated to be the true height. And for this, we used a root mean square error. And so just to kind of let you know what you're looking at here, along the y-axis, we have root mean square error. You want that to be low. The lower it is, the more accurate you are predicting environments. Here are along the x-axis, and then we're fasted by reps, and then by population type, so F3 versus real. And the point of this that I wanted you to take away was that you don't really see an increase, increasing your reps from 2 to 3. And so then I took that and just did 2 reps and increased it all the way up to 25 environments, just to see what the gains would be if you increased your throughput significantly. And so just to kind of see how you would interpret this graph is if you, through manual measurement, could only phenotype in a single environment, at what level of throughput would you need to increase if you had varying levels of accuracy in your high throughput methods? So for example, if you knew your accuracy was about twice as bad in your high throughput measurement, you would need 2 environments. 5 times as bad, you would need 4 environments. And 10 times as bad, you would need all the way up to 7 environments. And that would just be to break even, so you would need to increase it beyond that to see a net benefit. And so depending on your program, it might not be a benefit to you at this time for you to make that leap, but in some cases it might improve your program significantly. And this is in case you say, well, Holly, nobody ever tests the line in one environment. This is what your trade-offs would be for 2 environments. Any questions? So then we also looked at single marker analysis, and this was really detecting our ability to find QTL. And then I also looked at how many of these QTL we're detecting are not real. And for those of you not familiar, QTL stands for quantitative trait loci or locus. It's simply positions along the genome that contribute to the trait of interest. In this case, looking at, of those SNPs we generated, how many could the model say, yes, this is contributing to the trait, this is how much? No, it's not contributing to the trait. And so now along the y-axis, we're looking at proportion of QTL captured. So now you want this to be high. You want to capture more QTL and pick up more of the effects. And I know there's a lot going on here. So faceting along the x-axis is number of environments. So if you grew in 1, 2, or 10 environments, in an F3 or real population, real is on the bottom. And then along the x-axis is increasing population size. So in this case, before where we looked at increasing environments as being your real net benefit, in this instance, you can capture more QTL if you increase your population size. And so just to kind of parse 2 of these out, we're going to look at the least resource intensive method, which would be a single environment, only self to an F3, versus 10 environments of real population. And so as you can see, once you get to 10 environments, there's not a real difference that's made if you decrease your accuracy. So the accuracy becomes less of a factor. However, if you're going in a fewer number of environments, there's a lot of separation. So if your accuracy is not that much worse than your manual measurement, simply increasing your population size would significantly improve your QTL capture. And so next we looked at false QTL detection. So of the QTL detected by each model in each scenario, what yes, the SNP is contributing when the SNP was actually not contributing to the trait. And so what I thought was really interesting is that you don't really see much separation between accuracy types. And so what I took away from that was that increasing the accuracy of your measurement does not really do anything to decrease the risk of false QTL detection. The only way you could reduce your risk was to increase both environments, but mainly to increase population size. And even when we look here again at 1 versus 10 environments, even in the most in a real population, you're always going to have about 10% of the QTL that you think you're capturing not really have any effect on the trait. And so there are some advantages of high throughput phenotyping other than just accuracy, though some of the literature suggests that using a drone to measure types may actually be more accurate and more objective than manually measuring. You can also have the added temporal dimension. So maybe you used to take hand measurements for terminal plant height at the end of the season, but if you use a drone now, you can fly every week, and then you can extract a growth curve and have sort of this added dimension to your phenotype. And then you can also use a drone to capture phenotypes that you can't capture manually. So something like NDVI or other vegetation indices. However, there are some limitations to the simulation. It was relatively simplistic, so we didn't have any linkage disequilibrium or kinship generated between lines, and so that is a limitation that I just wanted to point out. And so back to our question, is data quantity or quality more important? Well, it depends. It sort of depends on what your goals are and how you can reallocate your resources. So if you're looking just to find kind of see the true genotypic value of a line, you should aim for more environments. But if you're trying to parse out small genetic effects, then you should really aim for a larger population size. However, I will say I think in general, quantity was more important than quality, with the caveat being as long as your quality wasn't so bad that you couldn't put enough resources into it to increase your throughput to overcome it. So the main takeaways, throughput is more important than accuracy, in my opinion, based on this data. Most readers probably already know that increasing the number of environments is very important, but I like to point that out because I think it means that our simulation is performing realistically. And then also, small effect QTL, as others have pointed out, can be really difficult to pin down, and increasing throughput reduced the false positive risk, but increasing throughput or accuracy did not eliminate this risk. So I will say that the value in QTL from that perspective for me would be their predictive capacity. So I would make this argument more towards genomic selection rather than chasing down QTL to try and find the genes controlling different traits. So overall, I think that as breeding programs transition to higher throughput phenotype collection methods, this will improve their breeding programs. That I'd like to acknowledge my lab, my committee, my funding sources, and I will take any questions.
So in your study, what did you call environments?
So we added, so I had a set number of environments, and then if a line was grown in that environment, it was given that environmental effect.
But by environment, are you talking about Oregon and Canada and
Right.
Texas, or are you talking about Brazos falling on one side of the road and Brazos falling on the other side of the road?
Right. So I had these effects were randomly generated with a uniform distribution or a standard distribution. And so some of the effects were big, some were small. And so we weren't really necessarily trying to figure out how different each environment was. That was something that was sort of being randomly generated to an extent.
So I think I missed it. When you say like 10 environments, we have one curious resident, and with the other 9 kind of that.
I guess I don't. So when you have one environment that has an erroneous measurement for a genotype or just in general?
Yeah. It's a gene that were high frequency. But then you have the right one for remaining 9 ones.
Right.
When you use all of that for, let's say, kilograms, within the other 9 characters.
Right, and so that's, I think the argument here is if you have more environments, then you can sort of overcome if you've had.
So I like what I'm trying to say is how can you make the point argument?
Right, well, so this is if you had 10 environments and the quality was poor across all your measurements. Because you have such a high quantity of data, it allows you to still parse out the genetic values.
You know, anything. Other questions? Okay. Thank you, Holly.